in business. All right, thanks everyone for coming to seminar today. Great to see a good turnout. It's my pleasure to introduce our speaker today, Dr. Aaron Carlisle from the University of Delaware. And Aaron and I go way back. We've known each other since undergrad. We both went to Princeton, and I kind of followed Aaron around a little bit in his journey. So everyone at Princeton has to do an undergraduate senior thesis. And we ended up having the same advisor. Aaron was a year ahead of me, and he came and spent time at the Hopkins Marine Station, took the kelp forest ecology class, diving around. And our advisor, Tom Hahn, had spent some time there, even though he was a bird guy, and said, hey, there's this really cool little snail, this little dog whale, it's named Necerius mendicus, super cool. Some of them have hydroids on their shells, some don't. Why don't you find out why? What is it? What's going on with these guys? And so Aaron, in the process of that class, and he did some of his own experiments, really testing whether there was any sort of effects on sea star predation. You know, do the little snails with the hydroids survive better than the ones that don't? And so then I came along a year later, and Tom's like, look at this cool senior thesis that Aaron did. Why don't you go and spend some more time studying these snails? And so I did. I decided to take it into the field, which was kind of a bad idea because I did tethering. That was a big mistake. Don't do tethering experiments unless you really have to. You basically, sorry, Ailish. Yeah. You just untangle things underwater all day long. That's basically it. So after that experience, then Aaron came here to Moss Landing Marine Labs where he did his master's. He was one of the first students in PSRC, the Pacific Shark Research Center. He did his master's with Rick Starr, Greg Kaye, and Dave Ebert, I'm sure, was involved there as well. And Aaron did a lot of work in the Elkhorn Slough, studying leopard sharks and their movements, doing acoustic tagging and tracking, and looking at seasonal movements and habitat use. And so that's when he added one of the first major skills that he continues to use into his toolbox there. After he finished his master's, then he went back to Hopkins, so to Stanford. He got his PhD and spent more time there. He worked with Barbara Block in her lab and was involved with a bunch of the cool tagging and tracking they do. You know, any sort of big, charismatic, pelagic, mysterious thing, like let's go put some tags on it. But there he really specialized and developed another tool that he uses, which was using stable isotopes and chemical tracers as a way to study their trophic ecology, ontogenetic changes, habitat use and movements and things. And even he came back to Moss Landing quite a bit. I'd see him playing with the micro mill and milling out little tiny bits of vertebrae from salmon sharks and white sharks, and then using that to kind of study their ecology and their trophic ecology over time. And then, you're living in Monterey. Why would you ever leave? So he stayed around for another 5 or 6 years, did a postdoc continuing on, at the Block Lab and all their cool, tagging, tracking, whatnot. He added in some genetic stuff, dabbled a little bit, a bunch of cool spatial modeling, and that's basically what he does today. So then in 2018, he moved to Delaware, got a position as assistant professor in fisheries ecology there, and he runs the Tracers Lab. Trophic, and Spatial Ecology Research Lab, but it's not Tracer with a C, it's with an S. Yeah, very creative. And so he does, basically he uses all these same tools in his lab, right? He uses biologging, electronic tagging, he uses the chemical tracers, stable isotopes, he uses the spatial modeling. He's just moved over there to the East Coast where there still is lots of cool, pelagic, mysterious, large, weird, interesting things that you can do this sort of work with. And again, a lot of his work's trying to study kind of what animals are doing, where are they, how are they doing it, linking it to physiology, ecology, and then also trying to have some of his work be really relevant for conservation and management, many of those species as well, especially in a changing climate. And so with that, so psyched you can come back to your alma mater and tell us what you've been doing. So he's going to be telling us about new biologging approaches for studying marine species and ecosystems. How do we do that is a good question. Mirroring, probably. Maybe fix it out of it and try to drag it over. Sometimes with students, that's...
What do you do?
Just try to...
Oh, there it is.
Now it's showing there. Okay.
So it was working. Nope.
This is the same thing with Ailish. Ailish, this is the same thing that happened at lab meeting yesterday.
Oh no, what happened? cats. I don't know if that matters. Okay. Yeah. All right. Okay. Wow, what an adventure, I tell you. So this is great to be back. Moss Landing is a really amazing place, and I hope you all appreciate it because you'll be out and move on, and once you move on, you'll realize how special this place is. And so this is the first time I've been back for a long time, and I really appreciate having the opportunity to come back and talk to you all. My wife is here, Maren. We actually got married on the right out here, right? So we have a long history with this place. We were actually here when it was built, and I helped plant lots of these plants when I worked in the benthic ecology lab. So it's a real trip coming back. So today I'm going to be talking about, like Scott mentioned, some of the work that I'm going on. I decided to focus on more of the biologging, so the animal telemetry tagging kind of work. But I have to, at least briefly touch on my history here. And so, a quick shout out to, here I am as a young person knocking out a leopard shark with Rick Starr. This is up in Elkhorn Slough. And I even built that little V-board there. I was very excited. You do?
Oh, yeah. Yeah, that thing's massive.
That's great. It's built to last, everyone. It was wonderful because I was in the ichthyology lab with Greg Kaye's lab, but Rick Starr was kind of my major advisor because he did acoustic telemetry, and wherever I go, I always do something different than what everyone else does, which is annoying but also fun because you get to learn a lot of things. And so, even just stepping out here, I was looking out and remembering, Arun's here, who was in my cohort back then as well, being out in the evening trying to beach seine gobies up in the Salinas River or whatever this extension is here, and there was this big question back then because the power plant was in training lots of juvenile fish, and one of the most abundant fishes they were finding were gobies, but no one ever saw them for the most part in any seines or surveys for the most part, and so me and another student, Chris Reinwald, we were working on a project, and I found this wonderful proposal. And it was an Elkhorn Slough mystery, the Great Goby Caper, or an estuary questionary. And we made this desperately seeking gobies thing. And this is our proposal for MS 103, which was, I think, marine ecology. Yeah. And so it was great. All these kind of hands-on field experiences are wonderful. I have a picture of Joe Bizzaro, me, and Greg Kaye. This is when Joe was heading off to the University of Washington. So, again, just a long time in this area. Spent 20 years in the Monterey Bay area. Longer than I've been anywhere else before moving to beautiful Delaware, which I'd never been to until I'd actually interviewed. So it's been a transition, but it's been a really fun time and a fun place to be. But so yeah, my lab is the tracer lab, and so we basically kind of have three main areas of research: trophic ecology, so what do things eat, what's their role in a food web; spatial ecology, so why'd animals go where they do, habitat use, things like that; and physiological ecology, more and more. Couldn't quite get the P into tracers, so we left that out. But yeah, so we do a lot of work, and for most part, we're doing a lot of different, mostly kind of focusing on the sharky side of things, but we do work on whelk, we do things on squid, and a lot of different species, so a little bit of everything. So the first thing we're going to be talking about is this new tag that we're working on, which is a way to kind of get at metabolic rate. Hey, everybody. Hi, no worries. So, all right, so I'm sure you all know metabolic rate, metabolism, all that kind of stuff. Everyone know that, right? The basics of it, right? So it's horribly complicated. You can see this kind of map of metabolic pathways that's, horrifying to think about, but I always kind of keep everything at the whole organismal level. But metabolic rate, or how much energy an animal uses to do whatever it needs to do, is really the most basic, fundamental physiological parameter there is. It drives everything in life and ecology, really. And it's very dynamic. It changes all over spatial, temporal scales. One of the big things that really impacts it is temperature, right? And so as things get warmer, metabolic rate gets higher, things like that. And there's a couple different ways that we can measure this. And so for mammals and terrestrial species, we use direct calorimetry, which is basically where you look at heat production of an animal. So you have food plus oxygen gives you carbon dioxide, water, ATP, and heat. So you measure actual heat production. If you're an aquatic animal, in water, it's specific heat capacity of water is immense, so you can't really measure temperature changes very well. So we use indirect calorimetry, which is food plus oxygen. So the oxygen consumption is what we use to estimate an indirect way of a proxy for metabolism. And so the way we do that is we use respirometers, which I'm sure you're all familiar with. Generally, you put an animal in a sealed box with water in it, and you measure how it uses oxygen over time. This is great because it's practical, it's pretty straightforward to do. You can control things, you can standardize techniques, you can control the environment, and we've been doing it for a long time. Some of the big drawbacks are, though, you can only really do it on smaller animals. It's stressful. Animals don't generally like being taken from one place and dropped in another and sealed up. It's not going to be normal behavior in the best of circumstances, right? In any circumstance, you're going to be off kilter, you're going to be stressed out, you can't really move normally. And in addition, you're in this very controlled environment. And so I also want to just make the brief point that we actually don't measure metabolic rate directly, right? That's what I mentioned, but we measure MO2, which is this indirect measurement, which is a rate at which you use oxygen over some unit of time per some unit of mass. So that's our proxy that we're working with. So there's several different ways of measuring it. Standard metabolic rate, equivalent to basal metabolic rate, so the cost of being alive. So for us, if you're asleep, you don't have any food in your stomach, that's going to be pretty close to your standard metabolic rate. No, just maintenance activities only, right? So repairing your tissues, doing breathing, whatever. But no growth, no activity, nothing else besides just being alive. So the way we measure that in fishes is we either knock them out, so like you can see with our little sandbar shark here, you anesthetize them, and then you can measure oxygen use in the water, or you can let them swim around little annular or static respirometers where they can move normally, and you look at different metrics of activity, and you can basically scale back to what it should look like if they're not moving at all. But there's a basic problem, which is that if you're dealing with a fish that moves around, when are you ever not doing anything? When are you not swimming? And when do you actually not have food in your stomach? And so when you have food in your stomach, who here has gotten the eating sweats or had a big meal on Thanksgiving, gotten sluggish? Your metabolism goes through the roof when you have a big meal. And so that's why we standardize our trials to fast animals. We don't have that specific dynamic action, which is the metabolic cost of digestion. So these metrics are kind of already a little bit artificial. So what we use is routine metabolic rate, which is where you put them into some sort of space where they can move around to some extent. They aren't anesthetized, they're undergoing routine activity. You can see here a tuna. This is a tuna in the TRCC down at Hopkins. This is another little perch or something in a little more smaller respirometer. But so this is kind of a better, more useful metric in a lot of ways. And so we throw a fish in the chamber. You can see on the y-axis you have oxygen content of the water and time on the x-axis. And basically you seal the animal on, you turn the flush pump off. The flush pump is recirculating freshly oxygenated water to keep oxygen levels high. And then you seal it up, and then you let the fish just draw down the oxygen, and you can get that rate over time. Then you turn the flush pump on, and you reoxygenate it. Then you have maximum metabolic rate, which is where the amount of oxygen that your body's taking up is maximized. You can't, basically your tissues can't get any more oxygen to them using, through cardiovascular delivery. So the maximum rate of uptake and delivery of oxygen to the tissues. So the way we do that is we exercise fish till they're exhausted. You can do that in a swim tunnel, like with this tuna, again at the TRCC, where you swim them until they basically start to swim back, can't maintain their position. Or in a static respirometer, you can use a chase method, where you basically take a stick and you chase them around or whack them until they get stressed out and flop over, and then they're stressed out. They're mad. Or you can just take them out, hold them in the water, expose them to the air for like a minute or two, and that's equivalent to this exercise into exhaustion. And so we do this so we can understand a lot more about these metabolic rates. And as I mentioned, temperature is really critical. And so I'm sure you're all familiar with Q10 values, which is the sensitivity of any kind of biological or enzymatic activity or process over a 10-degree change in temperature. And so basically for most, you can see in the upper right, you have teleost in the blue and the lazobranchs in the red. And so as you get temperature increasing, you get that linear increase. That's the Q10 effect. So if you go from 10 to 20 degrees, how much does your metabolic rate increase? And for most aquatic ectotherms, it's going to be in the range of 2 to 3. But it can vary a lot. And so you can see here, you have temperature in the left one, you have percent change in metabolic rate on the y-axis, and you have the gray line is Q10 of 3, the orange is 2, and then the blue is 1, means it's insensitive to temperature changes. But for elasmobranchs, it can be as high as almost 7. Bat rays, for example, one of our favorite little batoids here, their Q10 is about 6.8. So you can see that going into warmer temperatures has a huge impact on how much energy they need to use, just by being in warmer or cooler temperature. So once we understand these metrics, we understand the standard metabolic rate. You can see here you have temperature and the metabolic rate on the y-axis. As the red line is showing you that Q10 effect as things get warmer. The maximum metabolic rate, you get that black line. It kind of tips up. And you kind of measure the distance between those two metrics, and you get your aerobic scope curve. And where it's highest, that T-op, that's where the animal has the most excess scope to do everything else it needs to do: eat, live, grow, survive, reproduce, whatever. As you get further and further away from that, you get to the PGIS temperatures, which is where things start to kind of go bad for the animal. And then eventually you get to really hot or really cold temperatures where the animal's just going to die. And so when you think about the environment out there, as animals move through thermally dynamic environments, where they are in that aerobic scope curve is going to change, and that's going to really impact how much energy they have to do different things. But one of the big problems doing this kind of respirometry is that they always happen in these kinds of settings, right? And so highly controlled lab settings where we're controlling everything, we're fasting the fish. And the big problem, as you all are well aware of, is this does not reflect the ocean. The ocean is incredibly dynamic over space and time. So here's a map on the left of the Mid-Atlantic Bight where I am, and so you can see right up here, kind of up in there, that's Delaware Bay. Just within the bay, you can get huge temperature gradients. Seasonally, you can see on the right how quickly you have the Gulf Stream going up. Animals moving across that are going to have very different impacts, or their metabolic rate is going to be impacted in different ways. And it's not just spatial. You can see here on the bottom is a week in the life of a salmon shark. It's migrated from Alaska down towards Hawaii. And it's diving from the surface, kind of layer, down to 400, 500, 600 meters, and it's crossing these incredible gradients of temperature from almost 25, 26 degrees C down to 4 or 5 sometimes. That's going to impact their metabolic rate and energy use. So basically, everything we know is wrong. It's the best guess, but we don't have any idea of how this relates to what's going on in the field. And so this is a big problem, especially given that no one has ever measured what we call field metabolic rate, so the actual metabolic rate of an animal in the wild. We've never measured it for any fish. So they're the most diverse group of vertebrates in the planet. You know, well over half the biodiversity of vertebrates in the world are fishes. And we don't know what they're doing in the wild. So getting field metabolic rate, and this is actually true for any water-breathing animal. There's never been any water-breathing animal outside of like phytoplankton or anything like that that's had a metabolic rate captured and observed in the field. So what we've been working on is building a new tag that does this. And so this is still very much in process, but we've actually got it working pretty well, and I'm going to be talking a little bit about that today. So the basic idea is really simple. So you have water going into the shark's mouth, over the gills where oxygen extraction happens. So you get the shark's breath functionally, the gill exhalant that's coming out of the gills, and you measure how much oxygen's in that gill exhalant, and then you can basically, we're working on scaling it up to a whole animal metabolic rate. And so this is Andrea Leontiu, who's my first PhD student, who just graduated this summer. This was a lot of her work, and then Dr. John Cohen, who's the actual physiologist, who I'm just a fish guy, but he really has helped, these have been both of the two kind of really helping drive this thing. But so we're trying to develop this tool where we take these basic metrics, build a device you put on the dorsal fin using these newer technologies, fiber optic dissolved oxygen sensors, and stick it in the gills, and it basically logs all the oxygen consumption data of the gill exhalant, and basically you kind of pull together this very basic idea, very basic question, where you can estimate oxygen consumption if you know the oxygen saturation of the inhalant, the ambient water, the oxygen concentration of the exhalant, and then the flow rate, the rate at which the water is moving over the gills. And so this is one of the new versions of the tag. You can see on the right we're doing some flow testing. That's the wiggle stick. That's our speed sensor. And so we can see here in the bottom right, those are two oxygen sensors, Pyroscience Pico Subs. And so it has all these different loggers and electronic devices we basically stick in a package. So it's going to measure dissolved oxygen sensor of ambient oxygen, dissolved oxygen of gill exhalant, has an accelerometer on it to get tracks, accelerometry, magnetometer, gyroscope, things like that. Flow sensor for speed, batteries, data logger, and then a satellite tag for recovering the device. They put it out, and it stays on the animal for a few days, hours, however long. But short-term deployment, pops off, lifts the surface, and get it back. So how does this work? First thing we had to do was do a whole lot of captive experiments. We would go out and catch these adorable little baby sandbar sharks. And so the first thing we have to start to really be able to quantify is the amount of water that's moving over the gills. And so we measure gape. So you can see here, we put a little mouth guard in the mouth of the shark. This shark's anesthetized, so you can then take some pictures of it. And we basically anesthetize. We have that water flowing over the gills. And you can see that we have two of those fiber optic probes coming down. Is this actually a pointer? Somewhere? Does this have a laser pointer, or am I making that up? Doesn't seem to. Well, whatever. You can kind of see here. You have those two little fiber optic probes right there, and there. And so you're measuring the amount of water that's going in, the oxygen content of that. As it goes into the mouth, there is not all the water that goes into the mouth makes it through over the gills. There's a reduction in flow as it kind of is moving. There's like a bit of a bow wave. So we can't just go from a raw measurement of water out of the pipe. We have to figure out what is this reduction in flow, actual branchial flow that is moving over the gills, and then getting those two oxygen measurements. And so, this is some preliminary data I just got a week or so ago. We've been doing experiments. This is in our swim tunnel respirometer. And so a little sandbar shark here, and what we're doing is we're ramping up speeds and making it swim slower or faster, because the gape of a shark isn't always the same. If they're swimming faster, it might be lower. If they need more oxygen, they're going to open it more to get more oxygen moving over the gills. And so here on the upper right, you can see some of the preliminary data. The orange bars, or the kind of pinkish bars, whatever color that is, are animals moving at a slower speed, about 0.6 body lengths per second. And the green ones are faster rates of movement, like 0.8 body lengths per second. And so you can see that's over about a 10-minute trial. We would go up fast, slow, fast, over and over and over, because this is going to replicate potentially more what they do in the field. And so you can see you get this kind of interaction between how quickly they're moving and how open their mouth is. We need to know this so we can understand what's going on out in the field. But you can see that it kind of switches out, where when they're actually moving slower, their gape is a little bit bigger, so there's more volume moving over there's more volume, at least in terms of the area, but because they're moving slower, the actual functional volume is actually lower than when they're moving faster. So when they're moving 0.8, they reduce their gape a little bit, and that basically is probably going to be because if they move too fast, it's going to blow out potentially their gills. Their gills are very sensitive. And so, but you can see as you move along, they start to basically, as a shark is exercising, sharks don't normally just kind of constantly swim faster and faster. And so you can see it's effective as it's kind of probably getting a little bit tired. It's keeping its mouth open more and more because its oxygen requirements are going up. And so this is the kind of stuff that we're trying to figure out, how do we actually incorporate into the tag. And so that's why this flow reduction is so important. And so the next thing we did was we basically measured oxygen extraction at all these different places in the gills, inside and outside the gills. And we found that basically the best that gives us the most consistent average value is in the middle of the third gill. And then we did some experiments where we had the fiber optic sensors on the sharks doing our thing, and then we estimated their metabolic rate based on the gill exhalant, and then we had them in a static respirometer at the same time, a traditional method, so we actually get the actual metabolic rate that we know using standard approaches. And you can see those dashed lines are the estimate of metabolic tank, and those are different trials from the gill exhalant. And so what we found is that basically the flow reduction, we were able to basically use sensitivity analysis to say what's the most likely flow reduction, and it ends up being about 38%, which is really close to the one other estimate, which Dr. Nick Wegner here, you can see here, who's actually helping with this, another important collaborator on this project. He found for mako sharks, it's about 33%, so it's almost the same. So we figured out where to put the probe. We got a good estimate of flow reduction, so now we can actually start to scale up these point measurements of oxygen extraction to the gill to the whole animal metabolic rate, or MO2. And so the next step was to figure out, can we actually put this in a tag and get some actual data on an animal that's not anesthetized and is moving around under its own volition. So we worked with another friend, Dr. Jennifer Wiffles, and went to Ripley's Aquariums, have a captive facility in Buffalo, New York. So we went there in the middle of winter, which was lovely. And we worked on this very special lemon shark named Houdini, who had a very sad backstory. He was in a tank happily, and a person jumped in there naked and freaked him out, and he ran into the wall and damaged his back somehow. So now he kind of spends his life like that. And he's been put out to pasture, and he only swims to the right. He can't turn to the left. So they were okay letting us test on him because they're never going to put him back into a big tank. So we went up there, we were ready to throw it out, we were very excited, and the minute we got there and started to put it on the fin, we realized that our mode of attachment wasn't going to work and something broke. So we had to make an on-the-fly adjustment, and so one of the aquarists said, why don't you just use one of our weight belts and strap it around the animal? And we're like, well, we have nothing else to do, so let's give it a try. Houdini had some thoughts about that. This is just some pictures of us doing all this fun stuff. But there you can see Houdini over there, and let me play this.
Here we go. He's going.
He decided he did not.
Oh, boy. Oh, boy,
And he
it's going
actually
to come off. Oh, it's going to come off. Oh,
very
he did
exciting.
It. He did it. He did it.
And there
Well
he
done.
goes.
Well done.
Right? And so that was our first trip.
Honk.
That was not a success, but that's okay. We learned from it.
All right,
We went
here we go.
back
He's going.
later on,
Oh, boy.
and the big lesson was,
Oh, boy, it's
try
gonna
to
come
build
off.
a tag.
Oh, it's going to come off.
Everything
Oh, he
you
did
want.
it. He did it. He did it. Well
So when you
done.
get into this
Well
kind of
done.
technology development, it
Honk.
makes sense to do it. Don't try to go to the end right away.
All right.
Very important lesson. So we went back, and here we are with our next instance. Then the attachment was working. You can see there he is, and we're giving him a nice little shove out, and off he swam. And he didn't care at all. He was quite happy. Unfortunately, our fiber optic cables are super long and they come uncoiled, but he didn't really care. And you can see him here, swimming around, just happy. And the tag stayed on him for about an hour or so. At that point, we pulled him back in. We took the tag off, and off he swam. And so here's what we found. And so this is the accelerometry in the top. These are the XYZ kind of axes. And oxygen saturation is on the bottom. So you can see it goes from 20% up to about 80 or 90%. And you can see right off the bat, with the oxygen saturation, there's huge variability over time, right? So this isn't like this kind of constant, steady oxygen extraction. Up in the top, you can see the change in acceleration as the animal was either sitting on the bottom, which is what Houdini really is a champion at, or those few minutes when he's swimming in a circle to the right. And so you can see they're color-coded on the bottom. Blue is sitting, purple is swimming. And so we got the data back. We were able to actually get real kind of live data from the animal as it was swimming. So it was a great kind of a demonstration of the approach. And then we looked a little deeper, and so this is, here he is doing his thing, sitting on the bottom, just buckle pumping, right? So they can, not all sharks have to constantly swim. He was sitting there very happily. He spent about 3 hours just sitting on the bottom while we're prepping things, not doing anything. So this is normal for Houdini. But so we could look at the video cameras. We had video cameras up everywhere, and you can see the shark is inhaling and exhaling. And as it's doing that, you can see this up on the top. This is showing you the gill state, basically as it's going up and opening, and they're basically drawing water in up and down. And you can see the oxygen profile at that same time below, and if you kind of overlay them, it matches up perfectly. And so you're seeing this dynamic of they're pulling in oxygenated water, freshly oxygenated, fully oxygenated water. It's in the buccal cavity for some period of time, probably starting to have that oxygen being sucked out, and then they expel it, and it's that bolus of depleted oxygen that's being pushed out. And so you can see then on the bottom right, it's showing that you can really identify the different behaviors based on just the oxygen extraction of the water. And so it worked. We were very, very excited, and we were like, okay, something's here, we're gonna get it to work. So we had to do a bit more, and so then we did the next step in validating this tag, and we called Remora. But we used kind of a free-swimming shark and a big floating respirometer in Delaware Bay. You can see our beautiful Delaware Bay water. You can see the tag there and the shark swimming in circles. And so this allowed us to use that as a big static respirometer to get that traditional estimate of metabolic rate while we're getting that. The tag is recording all the parameters it does, so we can see how well does it match. Are we actually able to get good measurements of metabolic rate? This was a lot of work. Oops. A lot of people out in the field doing everything. It was an all hands on deck, but it was great. We got a lot of good data, and here's just an example of what it looks like. And so these sharks, a lot of them also just seem to not want to perform when we want them to, so they would just sit on the bottom, kind of like Houdini. So like half of our sharks would just sit there, and the other half would swim really well. So on the left, you're seeing a sitting shark, and on the right, you're seeing a swimming shark. Again, those colorful figures on the middle are the accelerometer data. And again, just kind of a way we're using that triaxial accelerometer to really understand the movements and kinematics of the shark. And then we have the oxygen extraction from the tank in the black lines. And then we have the oxygen utilization from the tag in the red lines. And so again, you can see very quickly that what the animal's doing results in very different patterns of oxygen use, because one, they're swimming, one, they're not. They're using more energy, therefore they need more oxygen. And so we compare them. You can see these are all these different sharks, multiple trials. The green symbols are the tank measurements, the static respirometer. The orange ones are the gill measurements. And so when we actually measure them and we use our approach, we actually get really close to the same overall value. But we get this incredible amount of much more detailed and nuanced information that you would never get from just this kind of linear regression of oxygen extraction over time. So again, it works. We're like, okay, cool. Now let's see if we can actually get this out and put it in the field. So we went down to Florida this last spring in March. We worked with Dr. Matajamian at FAU. And we tagged a bull shark, you can see her on the right, with the first kind of more well-developed remora. And we tagged it down there in the Indian River Lagoon. And over about 12 hours, 14 hours later, it popped up in this creek, San Sebastian Creek. So bull sharks are euryhaline. They're one of the few sharks, I'm sure as you all know, that can go into fresh water. And so this was cool because this was a shark going from water that's in the mid-20s part PPT up to almost fresh water. And so when you think about the osmoregulatory, energetic cost of osmoregulation, you can start asking some really cool questions. And so these are the data we got. Unfortunately, the accelerometer did not work. It either failed or Andrea forgot to turn it on. I have my suspicion. I'm not going to tell you which one it was. But we didn't get the accelerometry data, which was a bummer, but that's okay. And so you see in the top, that's the temperature. The dark kind of shaded areas are the nighttime. The light is during the day. The top panel is temperature. The black one is pressure. The accelerometer was on the outside would have had a much better record, but this was still the pressure signal is going to be muted because it's inside that pressure capsule, right? But it still does change, so you can still get general trends and change in depth. And so you can see right after we tagged it, it went to the bottom, then it kind of moved up and just started swimming up a little bit shallower and shallower. And then the third panel with the green and the orange lines are showing you, the green is the ambient dissolved oxygen of the water it's swimming through, and the gold is the gill exhalant. And then the bottom panel is the actual utilization, so the difference between those. And so you can see there's a lot in here. And so you can see this, we broke into 3 sections. We have the section where it's in the Indian River Lagoon, swimming kind of north. The second section is when it enters the San Sebastian Creek. And then the last one is the San Sebastian Creek Part 2, which is where it did some different things. And I'll talk about what this means here. So what happens is the shark swam to the bottom, started moving up. We think it was probably hugging the bottom. The lagoon is very shallow. And it basically, you can see as it moved north, there was actually a very large cold snap that night. The temperature dropped like 20 degrees C. And so we thought the sharks were actually going to swim offshore. We were kind of freaked out. But it actually went into the river because the river is warmer, which was cool. So it was finding a thermal refuge. But you can see it's kind of going shallower and shallower as it got closer to the mouth of that creek. And you can see the oxygen content, or the oxygen utilization, percent utilization. So this isn't metabolic rate. This is just the oxygen utilization of the gills in terms of oxygen content of the water. And so you can see that it kind of goes up a little bit. So that shark is probably swimming into the current. So it's exercising fairly intensively. So its oxygen requirements are going up over time. And it moves into Sebastian Creek, which has a ridiculous salt wedge. And so they catch freshwater fish at the surface, which is at like 4 meters, or at the surface, and then about 3 meters deep, they're catching like saltwater fish. And so the bull shark hit that wedge and basically started doing some slightly different things. Went too far. So here we are in Sebastian Creek Part 2. You see how the temperature becomes incredibly variable. Again, we don't know exactly what was going on because we didn't have the accelerometer data, but you can see that it's moving across that thermal gradient, which is that fresh and salt, that pycnocline, right, the thermocline. So it's kind of bouncing up and down, probably sticking along, hugging that edge of the salt wedge. At the same time, it basically goes a little bit deeper, but you can see the oxygen extraction and utilization actually goes up, and it's actually about the highest we get of the whole track, and that's going to be some combination of the increased cost of osmoregulation. There's also in fresh water, sharks are less buoyant. They don't have gas bladders. They have a large oily liver, which one of the reasons we think that sharks don't go into fresh water is they're no longer buoyant. And so they have to swim more just to stay up in the water column. In addition, they're dealing with these temperature fluctuations. We talked about Q10 effects. And so you're going from 22 to like 17 degrees, which can be a pretty big change in temperature. And then, basically the shark, we think, because you can see how the depth goes down and the oxygen extraction at the bottom there goes down, we think that it kind of just slowly glides maybe along the edge of that salt wedge down to the bottom until it gets into the salty part of that wedge. And you can see that when it's gliding, the oxygen utilization goes way down, so it's not using as much oxygen. And then it goes up once it hits that lower wedge, the salty part of the wedge, and it starts swimming up, and that temperature becomes nice and consistent because it's staying in that body of salt water. So slightly lower energy use because they don't have to spend as much time osmoregulating, they're more buoyant, and they're in a stable temperature. And so these are all just the kind of patterns that we're seeing, but when you break up these measurements and you actually estimate metabolic rate, we get something like this. So these are those sections, metabolic rate, so MO2 is on the y-axis. You have the first section that in the Indian River Lagoon, you have Sebastian Creek in the middle, and then the second part when it's in the kind of in the salty part of the creek. Each one of those black points is an instantaneous measurement of oxygen, of metabolic rate. So every second we're getting a metabolic rate estimate. And that blue line is just a simple linear regression of metabolic rate versus temperature. And so what do you see here in terms of temperature going from 19 to 21? In the Indian River Lagoon, does this make sense? Can anyone tell me? Does this make sense based on what we were just talking about? What happens to metabolic rate when it gets warmer? What's that? It goes up. Yeah. So does this go up? It doesn't. It goes down. And so if you actually use the Q10 and estimate the estimated metabolic rate based on the Q10 you see from the last point and say, okay, based on what the Q10 effect is at the end of that regression, what should it look like at the beginning of the track or at that change in temperature? You get an opposite trend. It should be following that green line, right? So we're seeing the opposite, and you see that in pretty much both of those two sections. You see that in San Sebastian Creek as well, part 1. And so the other thing you see is a lot of noise, especially as you get into that place where the shark starts to hit that salt wedge. And so you would never see this, you can't get this any other way, right? So these are field data from a bull shark moving up and down this dynamic environment. And that's kind of we were super excited to see this because this is the whole point of this effort is we only get these data from these very controlled systems. And the amount of range you're seeing doubling and tripling the metabolic rate over scales of seconds, right? So that has a huge effect. And you can see in San Sebastian Creek, part 2, when it was in the salt wedge, in the salty part, basically everything starts to settle down and starts to look more like normal. And you get a much more consistent and lower energy use while it's in the salt water. So you're kind of seeing that much higher energy use in the first two because it's swimming actively in the first section, and then it's dealing with osmoregulation and swimming in the second. So, take home, we actually, on Friday, we put out another one. You can see here, this is on Friday in the bay. There goes another shark. Have not heard from it. It's the second time we tried this summer, so something's been going wrong with our last few deployments, so incredibly frustrating. But that's how it goes, right? And so anyone who does science will tell you that science never goes the way you want it to go. And it's really just you keep pivoting, you keep plugging away. And that's where our experience through this is like failure, 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 success, failure, 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 success, maybe 2 successes and a lot more failures. But you keep doing it. So we have a lot more work, and we're still working on that, and we're hoping to maybe squeeze in one more deployment before the end of the year. All right, so that's the first vignette. I really want to talk quickly about one other thing we were working on, which is this idea of using sharks as ocean sensors. And so this is something that we have a long history in this area. Dan Costa and others at Santa Cruz have been putting CTD tags on elephant seals for a long time. People have been doing it. It's revolutionized our understanding, especially of polar seas, like because the elephant seals live there. They can collect data when ships can't get there and do the stuff. So you can get amazing data when you start to use animals as ocean observing platforms. And so the stuff I'm talking about, we have a paper that just came out. You can see down here on the bottom if you're interested. But the general idea is very simple. You basically put these tags that record these fundamental oceanographic parameters, and the animal is out there doing its natural thing, and when it comes to the surface, it transmits data in real time that then gets passed back to the IU system, like, right, so Senku's here, Integrated Ocean Observing System. For us, it's Marico's, the Mid-Atlantic regional node. And so really most of this work has been done on marine mammals, air breathers, and then marine reptiles, both air breathers, but both generally highly threatened, endangered, lots of permitting issues, hard to work with. And in the Mid-Atlantic, we don't have very many, especially marine mammals like seals and things like that. So we were interested, we got money, we got funding to try to advance the idea of using sharks as ocean sensors. And this is something that the lab that we came from at Stanford, Barbara Block, has done some work with as well. And the big advantage is if by using these animals, you can see up here, this is the relative, the top left one is showing you Argo floats. Anyone familiar with the Argo program here? So it's like this big program where they throw out these basic like floats that record all these data, and they sink down to the bottom like 1,000 meters every few days and come up and down. They basically just float around the currents and transmit data. So you get pretty good coverage in the open ocean. You can see in the top right, and basically, so here, if you look at the top left, pretty much where it's kind of in the warmer colors, that's where you get the, kind of neutral, almost whitish colors. The blue's not much. But in the right, you're seeing where we've currently have a lot of data from elephant seals for the most part, and it's around the poles. And so what we're trying to do is let's start putting these on these animals that use other parts of the world's ocean. So you can see in the bottom left, all the different telemetry data sets we have for sharks and fishes and everything like that, showing that we can start to fill in the gaps of where we can't get these observations using other approaches. And so this is important for us in the Mid-Atlantic Bight, Northwest Atlantic, because hurricanes are a big problem, right? So we're in Hurricane Alley, and this is the middle of hurricane season. And in the Mid-Atlantic Bight, we have a very unique oceanography. There's this cold pool that gets set up kind of off of New Jersey and New York, and that's like a branch, a little blurb of cold water from the Labrador Current comes down, and so you get at the top, you get water that's super hot, and down at the bottom, it drops like 15, 20 degrees C. And so if you're a hurricane, you're coming up and there's a cold pool there, that's like hitting the brakes. The hurricane slows down. If that cold pool is dissipated, it accelerates and it gets stronger and stronger. And so right now, the way we get these data for our part of the world is through the Argo float you can see in the middle. We send out research boats, big research boats, which as you all know are exceedingly expensive, cost a lot of money. Or we use gliders. And so the gliders cost a lot as well, but they also have certain limitations. And so you can see here, for example, those are some of the ideas of where you really need to get an idea of the ocean heat content. Everything is going underwater. Satellites are great, but they just look at the skin. They don't tell you anything about what's happening at depth. So you need to have animals that are moving through. And so this project, you can see in the upper right, that's the glider picket line. And so Dr. Matt Oliver, you can see in the middle, and Carolyn Wernicke, who's a PhD student who's working on this. Matt Oliver runs as part of this program. He has these Slocum gliders we throw out running those picket lines. You can see the lines the gliders basically traverse during the hurricane season in the Gulf of Mexico, Caribbean, and the Northwest Atlantic. And those gliders are great. The boats are great, all those things, but they have some very significant limitations, especially during the hurricane season when we need to have lots and lots of data. These models are very data hungry. And so we wanted to figure out, all right, so if we're going to use sharks, the first thing we need to do is figure out what kind of shark can you actually use, because not all sharks do the same thing. Every individual is different. Every species is different. And so we basically went through the literature and we worked with our collaborators to get as many satellite tag data sets to look at a few different basic parameters. One is, how often do they come to the surface based on satellite transmission? And so this, for example, is a spot tag that's on a dorsal fin of a shark. This is functionally what we're going to be doing. Every time it comes to the surface, it's going to be transmitting to satellites. And so this is just to get positions, but we get an idea of surfacing behavior. And based on the quality of the data that we get an idea of how long it's actually at the surface, which the longer it's there, the more opportunity is to transmit data to satellites, because satellite coverage is not perfect. We also are looking at where they're going and how you move around the ocean, so we can figure out what are the kind of environments that they're going to be observing. So different people using these tools might be interested in different oceanographic features. So this species might be great at observing mesoscale eddies. These ones might be good at observing gyres. Who knows, right? So kind of get an idea of what are the relative pros and cons of different species of sharks. And then, well, how can we show the utility of this? What's the cost effectiveness of using this tag versus these other approaches? And so this, for example, is showing you some of the data. We have it grouped by kind of habitat, tropical, low temperate, and high temperate environments. Each one of those colors is a different species and showing you the average track duration, how long was the tag transmitting versus how many number of locations did we actually get from those tags. And so those gray lines are showing you basically a one-to-one kind of transmission rate, where if you get like one transmission per day, it should be kind of around that line. So you can see, especially as you move over to the low temperate and high temperate, you get some of the animals that are above that line, which means they're actually doing a really good job. They have a lot more potential to provide us with more data. But more transmissions doesn't mean more data necessarily, because it might just be briefly hitting the surface. And so all these transmissions have a location class or location quality, an LC. So in this color-coded kind of strip chart here, the brighter colors, the yellows, are high quality location, meaning the tag is at the surface for a long time, where you're going to have more time, more bandwidth for the tag to transmit. Darker colors, blues, means they're coming to the surface, but they're not staying there very long. You're getting kind of crappy positions, so less opportunities to get data. So for example, you can see here, you have good, the Pacific mako, short fin mako is going to be pretty good, but the Atlantic tiger shark is not going to be terribly good, right? And so these are the things, you don't just want to go throw these tags out on anything. You need to have some idea of what to target and what to put them out on. This is complicated, don't worry too much about it. The basic idea is here is you can look at days since the tag was deployed and the number of transmissions like before. And so basically it's saying, well, how often can you expect a different species to give you a transmission? And so the little dotted line up here, SES, that's a southern elephant seal. Again, air breathers are great. They have to come to the surface to breathe, so you're going to get good data. That's kind of the gold standard. So the closer a line is to that elephant seal, the better it's going to be. And then those different dashed lines, the T 6 hours, T 12, that's going to say, like, that's what you'd expect if you're getting one transmission every 6 hours, one transmission every 12 hours or 24 hours, so on and so forth. And so you can see in the tropics, things like Galapagos sharks, gray reef sharks, oceanic whitetips aren't going to be very good, but the tiger sharks in the Pacific are going to be great. And then if you look over on the right, you start to kind of get an idea of, okay, blue sharks, common threshers are maybe going to be good. And the bottom is showing you the same thing, basically, but the general idea is you can start to use this framework to understand who do you put these tags on, because again, the tags aren't cheap. They're $7,000 or $8,000, which is a lot, but still has a lot of benefits. So you want something kind of in that, or that kind of triangle, right? That's going to be the kind of sweet spot. And then in terms of the types of environments they're going to be sampling, this is just some ordination. You have environmental. What are the kind of environments they're sampling? And so you can see you have your different loading factors in those blue lines. The diff ZL is the euphotic depth, so how clear is the water. The median SST is in the left kind of arrows going over there. You can see chlorophyll, difference in chlorophyll, so gradients in chlorophyll and gradients in sea surface temperatures. So for example, if you want to go somewhere with high chlorophyll, you would go for Pacific juvenile white sharks, for example, or something like that. Movement behaviors are going to impact this. So if you have animals that move fast and straight, they're going to have a very high straightness index. Other ones are going to be used first passage time, so how wiggly are their movements, and these are all going to impact how these animals sample the environment. And this is just a Stommel diagram, which for oceanographers, you should be familiar with this. Just an idea of the spatial and temporal scale of different oceanographic parameters, right? So you have, in the orange, you have eddies and rings, so like mesoscale kind of features. They're going to last, on the scale of months, and they're going to be, tens to hundreds of kilometers in scale. Then you have hurricanes, which are the kind of gold, orange, yellowish kind of color. Upwelling events for like California are kind of the blue. So on. Zooplankton swarms, right? So those very small scale dynamics are in that scale. And so the different ellipses are showing you where different sharks sample. So you kind of get sharks by using sharks, these animals as ocean sensors. As my graduate student Caroline says, we give them a backpack and a job. They're crossing all these different other traditional kinds of sampling devices. So if you look at the different dashed lines, those are the Argo floats, gliders, and ships. And so sharks kind of cover everything. And so they kind of aren't limited in the same ways as these traditional things. So then, getting to the last point of how effective is it? So these gliders, again, you have your glider picket line. They cost up to $1 million a pop. Every time you put them out, they cost tens, maybe $100,000 per deployment. They only stay out for a few weeks. And the data are also spatially and temporally autocorrelated. They throw away 95+% of the data because it doesn't give them new information. The CTD tag costs about $8,000. The sharks move really fast. So every data point you get is going to be new and give the models new information. And they last for a long time, months, if not even maybe years. We have no idea how long it's going to last yet. And the data are not autocorrelated. So in the left, you can see those are some blue shark tracks, for example. So you can imagine if those are collecting CTD profiles, you're going to be getting data over large swaths of the ocean. And that's just, so basically when you break it down, you can get a profile for each profile for a glider costs about $2,000 and about $300 per profile for shark. I'm going to skip over this because it's just more of the same stuff, and I'm pretty much done. Anyway, this is just showing you can actually get vertical stuff as well. This is from the first CTD that we ever put out on a salmon shark. And these are some of the profiles, and basically it showed that you can identify very unique different water masses vertically and do lots of very fun oceanography. But again, as a shark person, I just want to give the data to the oceanographers so they can do what they need to. So we think this is a really exciting kind of idea. This is just from this paper that I had a citation on earlier, showing you the relative, if we had tagged all these salmon sharks that Chris, Pearl, and I had tagged over our time at Stanford, if they had been collecting these CTD data, you can see some ideas of how much data we would have gotten and how that would have added to our understanding versus the World Ocean Database, WOD. So it really would help fill in all of our gaps. And this is just some video. We just put out our first CTE tags. This is actually, we put a tag on a white shark, a juvenile white shark. You can see it's moving around all over the place, doing great. And then we put our first one on some makos, seen right here. We put out 2 on makos, and they were doing great. They started being all over the place. We did the tagging off of Ocean City, which is right about here. So this is where the sharks were going. Then they went quiet after a few weeks, and we got very sad. But then one got caught off of Nantucket a few months ago, and the tag was still on it, but we weren't getting transmissions. But then we got a random transmission later in that day, but we haven't heard from it since. So we don't really know what's going on, and we're trying to figure out what's happening. So again, failure, failure, success, failure, 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 failure. But we're hopeful. So stay tuned. Hopefully we'll have more information. Hopefully we'll be putting these out at scale more for the next hurricane season. So with that, thanks for listening. I probably talked a bit long, but thank you.
All right, we have time for questions. I'm going to pass you the microphone. It's not to project your voice. It's so people online can hear your question. Question. You're closer.
All right, so amazing talk. I have a 2-part question. So one pertaining to the Remora tag. So I'm just curious, how much does it cost for you to make it, and have you lost it? And then the second part question is, it looks like that you put the Remora tags mainly on like young of the years, juveniles, and I'm assuming like based when sharks come inside the river or the lagoon, based off age, they have different thermal cues, I'm assuming. So have you thought about looking at different metabolic rates for adults and then younger years?
Yeah, no, this is, we want them to actually go on bigger animals, and so that's absolutely, it's going to change. And actually one of the things that we think is driving those disconnect between the Q10 and the observed data is thermal inertia. So as you're bigger, your whole body doesn't change temperature instantly. And so it takes time. Your body temperature has to adjust. So that's probably part of it, and no one's really thought too much about that. So we're scratching our heads about that right now. But yes, we've lost several. As I just mentioned, I think we've lost 3 this summer. Every time is heartbreaking, but hello. But yeah, it's a constant. Whenever you try new things, you're figuring everything out new every time, right? And so every time we learn a little bit, we get better. But the cost is probably about $6,000 a pop or so. And so for a PAT tag, those traditional kind of satellite tags that you put out and they pop off, those are about $4,000 a pop. So it's not that much more expensive than a lot of the other satellite tag technologies.
Okay, I'm going to go to Duncan while I do that. Have you thought about putting salinity tag on the remora so you can actually get
that's next. Yeah. And it's like the sensors, the loggers, it's all about size, right? And that's where the bigger the animal, the more stuff you can put on it. And so that's going to be something we're actually working on. We have a few people who are getting some pretty small salinity sensors we can put on. Yeah.
So that was a super cool presentation. Thank you much. One small off-topic question for my actual question. In your maximum metabolic rate slide, the left-hand picture, is that Dan Creer's thesis photos with cobia and Virginia? Yeah, I did my high school internship. I recognize that exact
cool.
goal.
Yeah, absolutely. Yeah,
The cobia
he and
boat.
Kevin Wing.
So,
Yep.
Small
world.
Weird, small world.
Yeah, because that was actually done just down the road from us. Yeah, at Wachapreek, a lot of it, right?
Would have been Gloucester Point.
What's that?
Gloucester Point.
Okay, Gloucester Point?
Yeah.
Okay. The sandbars were Wachapreek, I think, but it doesn't matter. Go on.
So I'm really curious with the metabolic rate tracking, have you observed any feeding events?
In the which tracking?
When you're tracking metabolism in these animals, have you observed feeding events?
The only data we have in the field is that one bull shark tag. So, yes, we should be able to see it, and I think the accelerometer would show it, but again, our accelerometer isn't on. And, you should see those SDA effects. So you should be able to see, like, after, if there's a big kind of jiggly thing where there's a lot of movement, and then you see this kind of peak, then you should be able to, that's going to probably be a pretty good indication of a big meal and a specific dam action. So you should be able to see it, yeah.
And have you considered putting like a urea type sensor and watching for alkaline tides coming out of the gills?
So getting those data is a whole other kind of ball of wax, and the sensor, it's all about sensors. And so we can only do this relatively recently because these new fiber optic sensors were developed in Germany. And so, again, people have been thinking about this stuff, but the tools have never been there. And so we just, when we started this, I was kind of looking at it, and no one had ever done it. And I was like, I don't know how to do it, but it seems like a good idea. Let's figure it out. And that's one of the reasons probably why things aren't working, because I don't know what I'm doing. But I'm learning. We're all learning. So, yeah. But, I mean, the more sensors, that's the thing. Once you get a big animal, you can put up a lot of stuff on it. And that's where it gets really exciting. Yeah. You can start putting stomach tags, different physiological metrics, things like that. Yeah.
Aaron, you might schedule a trip to Peru to see how they load down llamas as an example of how much stuff you can put on an animal.
Got it.
Great triumphant alumni return. It's good to see like the good guys win, honestly. So I enjoyed that. The tiger shark, it looked like it had a very different surface profile in the Pacific and Atlantic.
Yeah.
Is there
That's
a reason for that?
And that's one of the things that was most interesting to us looking at this, is that you can't assume the animals are doing the same thing everywhere. And we see the same thing with thresher sharks. In the Pacific, they actually come to the surface a lot. In the Atlantic, they don't. And
More
so
sunshine, just better out
yeah,
here.
it's just different environment,
Yeah.
right? And so it's like water column profile characteristics, temperature, it's all different.
Thanks. I should second what Joe said about seeing a good guy be successful in science. It's great to see. Glad it happened to you. Plenty of people are out there estimating field metabolic rate just from tail fin kicks and just making some assumptions about oxygen consumption. And I mean, seemed to be an opportunity here to validate some of those assumptions. And I'm just wondering which direction you think that's going to go.
So ODBA. Yeah.
You said it, not me.
Body acceleration, yeah. No, that's exactly true. But those are all based on calibrated swim tunnel studies, right? And so those are limited because of all the stuff we've been talking about. And so and the people who do that will tell you that, like, I don't even know if I can they feel comfortable extrapolating to a species, just an individual. Like, it's so variable. So it's really hard to do, but using those indirect metrics from the acceleration data, linking that to oxygen extraction in a swim tunnel, that's what people are doing right now. And that's great probably in some situations, but again, because of these limitations, it's going to fundamentally be biased in a lot of situations, and probably in a very narrow subset will it actually be at all accurate. That's my feeling. But we'll find out. Yeah.
Who else has any questions? Do we have any online, Misha? Nope. All right. Let you off the hook.
Well,
I think
thank you
people
all.
want to come up and chat some more afterwards. So thanks, everyone.
Yeah.
Thanks our
Thank you.
speaker.
I'm happy to chat, so if anyone has questions, just come on up. I won't bite your head off, I promise. Yeah, it's pretty much just a litany of failure after failure after failure. I know. It's